Live cell microscopy allows researchers to study fission yeast nuclear dynamics in mitosis and meiosis in real time. The strength of this method comes from studying nuclear processes in living cells under normal physiological conditions, which eliminates the need for toxic fixatives and stains.
This technique addresses questions related to protein timing, stability, and mobility that may not be amenable to genetic or biochemical manipulation. It is critical to pay close attention to the health and fitness of fission yeast cells before imaging. Always examine morphology and growth characteristics to ensure consistency of results across experiments.
This protocol shows a relatively simple way to prepare microscope slides that, when mastered correctly, can enhance the consistency and reproducibility of prolonged live cell imaging.
To begin, pick up cell matter from a cryogenic wake-up plate, streak it onto YES plates, and incubate them at 25 degrees Celsius or 32 degrees Celsius, depending on the yeast genotype, to wake up fission yeast strains from cryogenic preservation. Then, use a sterile loop to pick cells from individual colonies in the awakened fission yeast strains and inoculate them into test tubes containing 3 milliliters of YES liquid medium. Place the tubes in a shaker at 150 to 220 RPM to grow at 25 or 32 degrees Celsius overnight to late log phase, with a desired optical density reading of 0.7 to 1.0. Transfer 10 microliters of the cultured